Since the discovery of its major pathological hallmark, the senile plaque, Alzheimer's disease has seen an increase in diagnosis. Today, there are 5 million Americans living with Alzheimer's disease. Because of that, as you can see, there are many care and support facilities that have sprouted across the United States. However, the growth of the disease, which is to triple in the next 35 years, calls for resources that far exceeds what we currently have. By better understanding the structure and dynamics of the constituents of the senile plaque through a novel procedure which purifies the components of the senile plaque, I hope to further the field of medicine in Alzheimer's disease. To study the structure of the constituents, I could not use standard X-ray crystallography as the proteins are intrinsically disordered. A still picture of a continuously changing protein would be useless. Instead, I looked at the part of the protein that never changes, its atomic structure. To characterize the atomic structure, NMR continues to be the best modern method. For this, I found out that protein needs to be in monomeric form. However, the constituents of the plaque clump together so quickly it is hard to keep them in monomeric form when produced recombinantly. Therefore, many scientists had given up the thought of ever producing amyloid beta recombinantly and had given into synthetic amyloid beta, which is unphysiological and unnatural. My protocol is the first ever to make amyloid beta from the beginning to the end in recombinant form. It is 96% cheaper than any synthetic version and is the best method to go forward for NMR analysis of the atomic structure of the constituents of senile plaques. First, I grew my stability-tagged, histidine-tagged amyloid beta 42 in E. coli cells in OD2N15 labeled buffer so that my groups could be eventually picked up by the NMR. Then I changed transport buffer from guanidine hydrochloride to urea to help prevent aggregation and to help with the costs. However, the supernatant came out thick and I had to change my FPLC column to a nickel column to retrieve amyloid beta. With the literature base I got from solving my previous problem, I made changes efficiently and confidently. Instead of spending money and time life-lifingly spending with TeV buffer to cut off the fusion protein, I decided to use a simple dialysis. Next, to separate the components from the mixture, I used an HPLC hydrophobicity-based column. However, I decided to change the column from what was suggested. I changed the CA column to a more discrete C18 column. This produced protein in higher monomeric quality. I also changed the gradient on my HPLC to better change the components. My 1D FID results show high monomeric content, and my HSQC of amyloid beta 40A2T, which is the only mutant to prevent Alzheimer's disease, can be used for drug simulations. The reason I picked amyloid beta 40A2T was to test two of the many hypotheses out there due to the methods lacking to answer or validate them. I looked at a hypothesis calling for amyloid beta 40's inhibitory role in amyloid beta 42 aggregation, and A2T's inhibitory role in amyloid beta 42 aggregation. I found statistically backed results through NMR analysis overlays. This goes to show what the potential and what anyone can do with the protocol I have created. My research can be used in employing disease-modifying drugs, which defect the disease at its core, as opposed to the conventional use of chemical replacement drugs, which subdue the effects of the disease. I want to do this because chemical replacement drugs have performed at a subpar level in the past decade. In the future, I hope to better understand the role of aggregation of all the constituents of the senile plaque.
Thank you, God, Rensselaer Polytechnic Institute, Dr. Chunyi Wang, Ms. Nicolina Clementi, the Wang Research Lab, my parents, Mr. Nathaniel Colbert, and my siblings.